We talked about the human sex response before the break. After male ejaculation, I said that for the female, the fornix expands, and something I didn't point out there, you have a seminal pool right there. And then during resolution, when things shrink back, the cervix dips down into the seminal pool, and then sperm will have a chance to swim for fertilization. So that kind of takes us to the next chapter, fertilization and development. And this is chapter 28. Here's some basic terms. Pregnancy is defined as events that occur from fertilization until the infant is born. The conceptus is referring to the baby, the developing offspring. Gestation is the period of the last menstrual period until birth. Now the last menstrual period you weren't pregnant, but that's the thing that a mom can remember. And just add 2 weeks from there, that's when pregnancy began. Okay. It's about 40 weeks. The preembryo is the conceptus from fertilization, okay, to 2 weeks old. The embryo is 3 to 8 weeks. And we will spend most of our time in this chapter talking about pre-embryo, embryo phases. Fetal development, not so much, but a fetus is week 9 to birth. Again, we spend most of the time talking about fertilization up to 8 weeks. Okay. The relative size is shown here of the conceptus. Pre-embryo, embryo, a 12-week fetus. You start off as just a single cell. And that's where we start the conversation, fertilization. You start as a single cell, 2 cells come together. To accomplish fertilization, sperm must swim through all of these structures. The external os, the cervical canal, the internal os, the uterine cavity, swim through all the parts of the uterine tube and fertilize somewhere around here. The ampulla of the uterine tube is the typical site for fertilization, which I've talked about before. Now there are some pre-fertilization events that we talk about. First of all, ovulation. That is necessary for fertilization. That must happen prior to it. Pre-fertilization events. Ovulation is required, so I'll just put it there. And sperm can be viable for about 1 to 3 days in the female reproductive tract. An ovulated oocyte is viable for 12 to 24 hours. So that's about, if fertilization is to occur, copulation, ejaculation must occur no more than 3 days before ovulation or a day after. Okay, so that's about a 4-day window per cycle where you can get pregnant for each 28-day cycle. Now, in terms of the sperm must undergo what's called sperm capacitation. That's a softening of the spermatozoa head. That's 2 words, sperm capacitation. I'm just writing a softening of sperm head. The goal is to spill the lytic enzymes close to the egg so you can breach the barriers that surround the egg. Now, okay, these other terms, we'll get to it, but basically, that acrosomal reaction is a very important pre-fertilization event, but we'll get to it. Here, before the break, I taught you this structure, the AC. That must basically fuse and spill when you get close to the oocyte in the uterine tube. So, pretend this is in the uterine tube, and what you see here are barriers that sperm must get to. Okay, here's the oocyte, there's the cell membrane, but these two barriers that must be penetrated are, I'll say sperm must penetrate. Corona radiata. And zona pellucida. Those are not the oocyte, there are barriers around it. The corona radiata are the granulosa cells that were ovulated with the oocyte from the Graafian follicle. Corona means crown, so they're just cells that came with it from the Graafian. Now, the zona pellucida was secreted by the oocyte. They're glycoproteins. A glycoprotein eggshell secreted by oocyte. And the picture shows sperm getting in. This one got in. So this one, this sperm will basically allow the oocyte to complete meiosis II for conception. So a more complete picture of these pre-fertilization events. Is shown in this figure here. It's a pretty good figure. Let's call it pre-fertilization events, sperm penetration. Sperm must get through here. It's not fertilization, it's still pre. I'll read what it says here. Approach. Aided by enzymes on its surface. Aided by enzymes on its surface, the sperm weaves its way past granulosa cells of the corona radiata. So that's kind of what's shown there. That's the first step. Breach corona radiata. Or penetrate. One. Sperm penetrates. Corona radiata. Enzymes on sperm head help this. So that sperm gets to this white layer here called the zona pellucida. So this figure here shows what happens after that. The second step is called an acrosomal reaction. It's the binding of the sperm receptors to the zona pellucida, causes calcium levels within the sperm to rise. It triggers this acrosomal reaction. Acrosomal enzymes from many sperm, they digest holes through the zona pellucida, clearing the path to the oocyte membrane. So there's a picture of it. So what's happening is, step 2, it doesn't say it here, but sperm bind this protein called zona pellucida protein 3, ZP3. You bind ZP3 in the zona pellucida. That's species specific. The sperm did the ZP3 thing. You know, animal sperm wouldn't recognize it. But anyways, that triggers the acrosomal reaction. So it's all the reddish stuff, I guess. Well, anyways, when I drew it earlier, I drew a sperm head with purple for the nuke. And it's surrounded by a double layer of acrosome. I used green. And I said there's lighted chemicals inside there. And that was surrounded by a red plasma membrane. So when you bind ZP3, it triggers a reaction, you get a rise in calcium levels, and what happens is the double membrane, those will fuse, and the cell membrane will burst open. So maybe the cell membrane kind of like opens up. Here's a nuke. And then when the two green membranes of the acrosome fuse, you just kind of get one membrane, and you've released chemicals to digest ZP, zona pellucida. So that's this frame here. That allows the sperm to penetrate all the way to the oocyte membrane, which has sperm binding receptors on it. That's the next step. So binding, step 3. The sperm's membrane binds to the oocyte's sperm binding receptors. Sperm binds sperm receptors on oocyte membrane. There's a fusion there. And they fuse right there. And notice how the head pivots at the neck to increase the contact. Step 4 is the fusion. The sperm's membrane binds to the oocyte's sperm binding receptors, is what we just did. So the fusion that happens after that is sperm and oocyte plasma membranes fuse, and the sperm contents enter the oocyte. I guess they're kind of showing that. Well, they don't really show it. But the nuke is all that they contribute. So we'll write that down. Sperm oocyte membranes fuse, and the sperm donates the nucleus. I'll put sperm nuke enters. Although they're not really showing that there. So when this happens, a very important event happens by the oocyte immediately. It's called a block to polyspermy. You only want one sperm to fertilize. If more than one get in, you never make it past fertilization. You never make it to the first cell division, okay, of the baby. So this is very important. It's called, as soon as sperm gets in, get the nuke, you block polyspermy. You block the other sperm from getting in. Keep them out. Okay, so the block to polyspermy is, let's look at the picture. Here we see on the right, the sperm nuke got in, and that triggers a reaction. In the cortex of the oocyte, there's these granules. And then when the sperm gets in, it triggers this cortical reaction where the granules in the cortex release these chemicals that harden zona pellucida, and that's the block. Let me read it from the slide here. So the block to polyspermy is calcium levels rise in the oocyte, triggers the cortical reaction. It's an exocytosis of those blue granules. As a result, zona pellucida hardens, and the zona pellucida's sperm-binding receptors are clipped off. That'll keep all the other sperm out. So, the block to polyspermy is a cortical reaction where cortical granule, let me show the picture, they're blue. In this figure, they're blue. Cortical granules release substances that harden zona pellucida, and clip off sperm receptors. We have models that kind of show these pre-fertilization events. There's a model that is part of your study guide, and we kind of illustrated in the fertilizing. Well, it didn't quite fertilize yet. There's the sperm there. Number 1, that's a 1. I see cells here. I would identify that as corona radiata. The white layer, number 2, is zona pellucida. 3 is the oocyte. And 4 is the nucleus of it. They call it the ovo, which is the nucleus of the oocyte. So once the sperm nuke gets in, we're done with pre-fertilization. Now this is actual fertilization. I'll just drop the pre. Fertilization events. So what you see here, I always like to tell students, you know, as you go through these events, it's a process, and see how it's different in the next step. So take inventory of what you see in the first step here, events of fertilization. Do you see corona radiata? Yes, it's there. Check. I'll just list it. Okay, it was penetrated, but it's still there. Okay. So it's interesting to know. I think the misconception is people think the sperm that arrive first are the fertilizing sperm. Well, that's not true. Remember, in the ejaculate, you have millions of sperm. You know, by the time you get to the uterine tube in the apula part, you're down to 50 or 100. Okay. The first ones that get there aren't the fertilizing sperm. They're the ones punching holes in the barriers. Corona radiata, zona pellucida. Maybe the second wave of sperm gets in. Okay. Corona radiata is still there. Zona pellucida is there, the white layer. So I'm just listing them because they're present. Let's take a look at what's going on with meiosis. You're still in meiosis 2. Looks like you're in anaphase there. And I see a polar body, first polar body. It's there. Notice how the first polar body is outside the oocyte, inside the ZP, zona pellucida. But it's been ejected from the cell. It's kind of trapped in between. Okay, well, it looks like you're going to finish meiosis tube, right? I'll put oocyte. It's going to finish meiosis tube. The sperm finished already. The oocyte completes meiosis tube because it's not done yet. And what I did was, we have different models that look at this step in slightly different ways, and it kind of confuses students. Because we're all trying to show you the same thing, but different. So I thought it'd be good to show you all in the lecture. This is like the picture in the book, right? Zona pellucida, the white layer, corona radiata, I'm finishing meiosis 2, first polar body, there's the sperm nuke. That's the haploid DNA from the male. In this model here, they don't show you corona radiata or ZP, really. But let me ask you a question. Is this the fertilizing sperm, yes or no? No. No is correct. That's the tail of the fertilizing sperm, and there's the sperm nuke that got in. Okay. Look at the colors, blue, yellow, red, yellow. That is of the ovum. Here's the ovum. The chromosomes are illustrated as black dots, don't know why. The last number 3, the ovum. 2, oocyte. 1, zona pellucida. True or false? The sperm has contacted the oocyte membrane. True. I always ask that on the lab practical, so see if you can remember that. Yeah. True. The yellow is the oocyte membrane. He made it. He is the fertilizing sperm. That cell is. Okay, next step, events of fertilization. What happens is you form this male and female pronucleus. Okay? Male, female pronuclei form. That's basically what's happening here. And take an inventory. Do you see corona radiata? It's still there. It's going away. What about ZP? Yes. Yes and yes. And let's identify our polar bodies. These two are from the first one. Therefore, what is that one? Second polar body. Yeah. Okay, so we're starting with below. This is a weird situation where you have both sets of chromosomes, N and N, in one cell, but they're not fused yet. So I call it N plus N. That's just me. I like to call it that. I mean, you got one that's of the male. It's in its own little bubble, pronucleus, and then the female one, it's in her own. So it's like N plus. You won't really see that in the book, but you should know that. Because I want to distinguish N plus N before the zygote forms. And so what you got there is, well, let's move on. Here's another model that's close to what we just looked at, and I kind of rotated the picture to make it match the model that's in the classroom. Here's the N male from the sperm. Okay, here's the male pronucleus here. Looks like a little parachute. See the two red dots? I think that's the chromosomes from the male. I don't know why it's the two red dots. Go with it. Red dots, male genes. Here's the female. Okay, it hasn't quite formed yet. And it's in anaphase 2. So half of the dots are going to go with this. The other half are ejected as the second polar body, but not yet. That's the first polar body. Do you see how there's 4 dots? I believe the 4 dots represent the 2 polar bodies from the first polar body. So go with that. That's what we'll go with. All right, next step. The pronuclei, they swell and they get close to each other. And you can see the spindle apparatus forming. It's going to help in mitosis. Because right now, this is fertilization. We're just talking about one cell, but it must divide. So I'm writing male and female pronuclei swell and move towards each other. It's still N plus N. So we have a lot of models in the room that kind of illustrate this part, the N plus N thing. I took the picture, we just looked at it and rotated it a little bit, N plus N. But our polar bodies up here, okay, that one's the second one. These two are from the first polar body. This one here, the 1 and the 2, that's from the male. That's the female. Okay, here are the polar bodies. This is the second polar body. These 2 polar bodies are from the first polar body. So make a connection here. We're going to go back to the previous slide. See the red dots? And I said those were the male chromosome things. So in this model here, that's how they look. Okay. Now pink is usually associated with female, but this is not that. This is like, I think they made the model before that came about actually. This comes after that, because see how the chromosomes have condensed? And you're getting ready for cell division, that happens later. Okay. But still, in all 3, it's N plus N. Let's move on. And finally, conception. Zygote formation is conception where the chromosomes fuse. One cell stage. We all started life, hard to believe, as one cell. There's some cell in your body that was like the first cell ever, right? It's diploid. You've restored the full complement of DNA. It's no longer N plus N. They got together. When the chromosomes intermix, that's conception. That's zygote formation. Diploid. So a zygote is diploid. Do you see zona pellucida?
Yes.
Yes. Do you see corona radiata? No. Gone by this time. It's about a day. You know, all this stuff takes about a day. I see polar bodies stuck inside zona pellucida, chromosomes in the mix. Here's a real picture of it. I see a faint zona pellucida. I see the pronuclei fusing, polar bodies stuck inside. So basically, we have a model of a one-cell stage. Not much to look at, just one cell. All the good stuff's happening inside. The chromosomes intermixing. It's getting ready to become a human. So you got you're one cell, and you have to go through many divisions. And so to sum up, sperm enters. N plus N. Once you get the zygote formation, boom, that is the act of fertilization, conception, zygote forms. And you can start undergoing more development. But I think this is a good place to pick up next week. Yeah, question?
So is this all happening in one day? Or?
Yeah. Yeah, all this happens in about a day. Okay. Remember, you have a histology quiz on repro. It's on the schedule. I want to remind you. And I want to have that at the beginning of class. So basically, you're not going to have a PowerPoint quiz. I'll just give you the histology quiz, and that's it. Yeah. There's nothing scheduled for lab today. But if you want to stay and study the models, you're more than welcome to. If not, I'll see you Monday.